Hey, we're fearing citric acid cycle, and you can call us. Today we're gonna talk about citric acid cycle, and let's begin.
TC cycle.
Let's have a look at the names of chemical pathway that we're going to cover. Citric acid cycle, TCA cycle, and Krebs cycles. They're actually all equal. First, for the citric acid cycle, it is named because citrate is the first molecule formed when acetyl-CoA and oxaloacetate is joined together. Second, TCA cycle is an abbreviated term for tricarboxylic acid cycle. Moreover, many intermediates have 3 carboxyl groups. Third, Krebs cycles is named because a researcher called Hans Krebs had discovered this wonderful cycle. This chemical pathway is crucial in metabolism of cells because of the following points. First, it plays an amphibolic role in cellular metabolism. Second, it is the main hub of producing energy, such as NADH, FADH2, and GTP, that drives ATP production. Third, it releases waste material such as carbon dioxide. Lastly, it produces biosynthetic intermediates to provide building blocks. Citric acid cycle is so complicated due to tight regulation following 12 precise biochemical rules regarding cellular energy. But still, this pathway takes the form of a cycle due to the continuous regeneration of oxaloacetate, and the molecule is needed to bind acetyl-CoA to start the cycle again. Specifically, multiple oxidation steps to harvest high energy electrons and production of carbon dioxide, GTP and ATP, and NADH and FADH2 as electron carriers. Lastly, fine regulation and integration with other metabolic pathways too. In addition, this wonderful pathway is efficient and smartly designed. It is because it tracks a lot of energy from carbon fuels, for example, 3 NADH, 1 NADH2, 1 GTP or ATP per acetyl-CoA. This leads to about 10 ATP per cycle.
Now let's do a brief introduction of the citric acid cycle. First, we begin with acetyl-CoA, a 2-carbon molecule that combines with a 4-carbon molecule oxaloacetate to form a 6-carbon molecule citrate. Keep in mind that the enzymes here that are mentioned in the cycle are for the key irreversible steps within the cycle. These irreversible steps are major regulation checkpoints within the cycle to establish and commit the cycle into making and producing energy for the cell. So, citrate is then converted into isocitrate, which is another 6-carbon molecule, and isocitrate is then oxidized into alpha-ketoglutarate, a 5-carbon molecule. Here we release 1 carbon dioxide molecule, and then NADH, 1 energy molecule, is formed. This is the second irreversible step within the cycle. And then alpha-ketoglutarate is then converted into Succinyl-CoA, a 4-carbon molecule. And here we also release a carbon dioxide molecule and form 1 energy molecule as well. This is the third and final irreversible step within the cycle. And these three irreversible steps are catalyzed by these 3 important enzymes. So these are where enzyme activity is tightly regulated and controlled. From here on, when energy is made or formed, these are all irreversible steps, and the 4 carbon molecules are kept within, but these are all then formed into different forms. So Succinyl-CoA is formed into succinate, succinate is formed into fumarate, fumarate into malate, and finally generating the last important molecule, oxaloacetate. Oxaloacetate is the 8th intermediate, but is important to regenerate and power this cycle to continue and start cellular respiration, which provides energy for almost every process in our entire body. The products generated per each cycle of citric acid cycle is 1 GTP, 3 NADH, 1 FADH2, and 2 carbon dioxide, which is released.
Okay, now Mian and Nori will explain about the reactions 2 to 8 in an easy way. So the first step is from citrate to isocitrate via cis-aconitase catalyzed by the enzyme Acotinase. It is to reposition the hydroxyl group, which I will explain how. So first, Acotinase catalyzes a dehydration reaction, removing water without extra energy. It uses a iron-sulfur cluster where the 4th iron binds to citrate. This binding holds citrate perfectly and polarizes the COH bond, pulling electron density toward the iron, making OH a better leaving group. At the same time, a neighboring carbon loses a proton, and the proton and hydroxyl group combine to form water and leaves, making both carbons miss a bond. The 2 carbons now form a double bond, and voila, cis-aconitate is made. Next, cis-aconitate rotates 180 degrees in the active site for the next step. Acotinase now adds water back across the double bond, and the double bond acts as a nucleophile, attracting water. The oxygen from water bonds here, and the proton also bonds here, and double bond disappears, replaced by 2 single bonds, giving us isocitrate. So even though delta G naught prime is positive, the real delta G is negative in the cell. And why? Because the concentration for isocitrate is very low. It's constantly pulled away by the next enzyme, isocitrate dehydrogenase, lowering concentration and making delta G negative, pulling the reaction forward. So in summary, acotinase converts citrate to isocitrate through a reversible dehydration-hydration sequence using cis-aconatate. So I will go on to the next step from isocitrate to alpha-ketoglutarate through the enzyme isocitrate dehydrogenase. This step performs the first oxidative decarboxylation of the cycle, producing NADH, a key energy molecule. First, isocitrate is oxidized, which releases energy. The enzyme catalyzes hydride transfer to NAD+, producing NADH. This is a group transfer. Electrons are being sent to a more stable electron sink, NAD+. Now, because C2 only has 3 bonds, a proton from the hydroxyl group is removed, allowing a new double bond to form, and this creates oxalosuccinate, an unstable beta-keto acid intermediate. Next, the beta-carboxyl group is removed as CO2. Why? Because the adjacent carbonyl pulls electron density, making the bond easier to break. As CO2 leaves, its bonds remain and forming a double bond here. Now, carbon can't have 5 electrons, so the pi electron of carbonyl group shift up to oxygen, creating O negative, an unstable enol intermediate. Here, manganese 2+ plays a key role, stabilizing the negative charge and transition state by acting as an electron sink. Next, the enol rearranges through tautomerization, and the CC double bond breaks, electrons shift back to oxygen to form a new carbonyl, and C3 gains a proton, restoring a single bond, making alpha-ketoglutarate. So this step is important because NADH is formed, which will later donate electrons to ETC to generate ATP, which is energy. Let's go on to next step. This step is from alpha-ketoglutarate to Succinyl-CoA, very similar step from pyruvate to acetyl-CoA with the same cofactors, so you should go back and check. This reaction is catalyzed by a multi-enzyme complex, alpha-ketoglutarate dehydrogenase complex, using these 5 cofactors. Remember, this step is also an oxidative decarboxylation step. So first, TPP acts as a nucleophile and attacks the carbonyl carbon. This weakens the bond between C4 and C5, which breaks, and CO2 is released. The electrons move toward the carbonyl group, forming a carbon ion. The carbonyl group stabilizes this with resonance, forming an O negative, which is protonated to make the OH group. And this gives us TPP-bound hydroxyethyl intermediate. Now the lipoamine arm swings in and it accepts the hydroxyethyl group from TPP. The OH is oxidized to carbonyl, forming a succinyl group attached to lipoate. The succinyl group is transferred from lipoate to CoA with a free thiol group forming Succinyl-CoA. This is a high-energy thioester bond, and the energy is conserved to go to the next step. Now lipoate is in its reduced form. It's oxidized back to its disulfide form by FAD, which becomes FADH2. But NAD+ is like, I'll take those electrons, and oxidizes FADH2, restoring FAZ and making NADH in a proton. This final step has a strongly favorable delta G, helping pull the entire reaction forward. So in summary, CO2 is released, electrons move step by step, Succinyl-CoA is made, and NADH is generated. Next, conversion of Succinyl-CoA to succinate. Really important because it makes GTP or ATP without using the electron transport chain. This reaction is catalyzed by the enzyme Succinyl-CoA synthetase. So Succinyl-CoA contains a high-energy thioester bond, a carbon-sulfur bond. Breaking this bond releases a large amount of free energy. So first, inorganic phosphate acting as a nucleophile attacks the carbonyl carbon of the succinyl group because the carbonyl group is electrophilic, wanting electrons. This breaks the thioester bond, and phosphate replaces CoA, being a group transfer, Rule 7. This forms a new high-energy compound, succinyl phosphate, an acyl phosphate intermediate. Next, the imidazoles ring of histidine residue acts as a nucleophile. Its nitrogen attacks the phosphorus in succinyl phosphate, forming a covalent bond over here. This breaks the bond, transferring the phosphate to the enzyme. Now, the phosphate group on histidine is transferred to GDP or ADP, forming GTP or ATP. This is true substrate-level phosphorylation, making GTP directly from metabolic energy, not through oxidative phosphorylation. Here, even though delta G naught prime is about 0, the reaction can proceed in either directions, depending on whether the cell needs more or has enough of GTP. So again, this step is important because it's the only substrate-level phosphorylation in the TCA cycle.
Here is the next step. This is a step that succinate is oxidized to become fumarate. This reaction occurs to make subsequent reactions more efficient and to obtain energy in the most efficient way. In this reaction, the outcomes are fumarate and 1-FADH2. Since Rule 2 states that oxidation releases energy, succinate releases two energies with the help of succinate dehydrogenase, which is the enzyme that takes two hydrogens from succinate molecules so that it will make double bond between carbon atoms, and FAD plus will take these two energies to store and bring into the respiratory chain, and finally fumarate is formed. The next step is a step that fumarate will hydrate across a double bond with the help of fumarase to become elmalate. Again, the purpose of this reaction is to make subsequent reactions more efficient. The outcome is elmalate. Before fumarate is converted to elmalate, it goes through the carbanion transition state, where the double bond of fumarate is converted back to the single bond by a hydroxyl group, bonding to one carbon atom. And this H plus is bonded to the other negatively charged carbon atom, resulting in the formation of elmalate. According to Rule 10, loss or gain of water molecules and cis-trans rearrangement does not require external energy input, so this reaction will occur spontaneously. Here is the last step for the citric acid cycle. This is the oxidation of elmalate to become oxaloacetate, which is a substance essential for making the starting substance of this cycle. The reason for this reaction is again to make subsequent reactions more efficient and obtain energy in the most efficient way. The outcomes for this reaction are oxaloacetate, 1-NADH, and 1H+. When NAD+ takes two hydrogen atoms and gets two energies, which is the oxidation of elmalate, NADH and H+ are produced, and this follows Rule 2. Next, the delta G 0- value of this reaction is 29.7 kJ per mole, a very large positive value. Therefore, at first glance, it may seem like a non-spontaneous reaction, but as written in Rule 12, not only the value of delta G 0-, but also the concentration of reactants and products play a role in whether a chemical reaction occurs spontaneously or not. In fact, the concentration of oxaloacetate in the cell is very low, while the concentration of elmalate is very high. In addition, the free energy of the next reaction is a large negative value, and due to the action of malate dehydrogenase, this reaction occurs spontaneously.
Now that we've seen how the citric acid cycle depends on key enzymes and specific rules to keep energy production going, let's look at what happens if a certain essential cofactor is missing, specifically in a condition called beriberi. Beriberi? Nope. Beriberi. Beriberi is a disease caused by a deficiency of vitamin B1 or thiamine, an essential vitamin that your body needs to convert food into energy. Beriberi could result from a poor diet, alcoholism, or any conditions that block effective nutrient absorption. The vitamin thiamine is important for enzymes that aid in energy production, especially for energy-demanding tissues such as the nervous system, heart, and muscles. There are various symptoms of beriberi that fall under two types of beriberi, dry and wet, and the symptoms are as shown here. Ultimately, the worst outcome of beriberi, both wet and dry, if untreated, is death. Now, how does a deficiency of thiamine cause beriberi? Thiamine is used to make TPP, thiamine pyrophosphate, an essential cofactor for the two critical enzymes, pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. Pyruvate dehydrogenase converts pyruvate into acetyl-CoA. So even though pyruvate dehydrogenase is not in the TCA cycle, it is essential because it links glycolysis to the TCA cycle. So after a meal, glucose is broken down into pyruvate through glycolysis and is converted into acetyl-CoA, which is then to start the TCA cycle. Alpha-ketoglutarate dehydrogenase is within the cycle, and it catalyzes the conversion of alpha-ketoglutarate into Succinyl-CoA. And keep in mind, this is one of the energy-releasing steps that includes an irreversible step, so this is a major regulation checkpoint within the cycle. So even though TPP isn't a direct component of the Krebs cycle, it's required to keep the energy production pathway running smoothly and efficiently. So when there's a thiamine deficiency and TPP is lacking, pyruvate cannot be converted into acetyl-CoA, and alpha-ketoglutarate cannot be converted into Succinyl-CoA. And so these two accumulate, however, it cannot be converted. So this will slow or even block the TCA cycle, causing less ATP to be produced. Since the TCA cycle is a major source of energy, especially for high-demanding tissues like the heart, nerves, and muscles, these energy shortfalls leads to the symptoms of beriberi. To prevent and cure beriberi, it is essential to restore the levels of thiamine in your body. You could do so through supplementations, injections, or change of long-term diet. So, remember to eat healthy and stay healthy, because getting this disease is very, very bad.